Today we're talking about bacteria and viruses. We talked about how things are living and non-living, so this is going to be our example. Bacteria are living, viruses are not living. So in this image, we have a bunch of bacteria that affect humans. We have in the upper left-hand corner a individual who has been diagnosed with having a flesh-eating bacteria. I hope none of us come into contact with that. Number two is Swiss cheese is made with bacteria. Yogurt, on the side of your yogurt, you'll see live and active cultures. And in this one, it has S. thermophilus, L. bulgaricus, and L. acidophilus. Those are all different strains of bacteria, and they help us digest. And number four is anthrax. There are different types of anthrax, and this is a cutaneous, so a skin issue with anthrax. So bacteria are prokaryotes, which means that they are unicellular organisms that lack a nucleus. We talked about eukaryotes, so they have a nucleus. So those are the two types of cells. All bacteria are divided into two groups. They are kingdom eubacteria, which is domain bacteria. These live all around us. These are the ones that get us sick, that we use in our food. They have cell walls of peptidoglycan, which is a carbohydrate. So that's one way that scientists can know what group they're in. The other group is called Archaebacteria or domain Archaea. So if you know the word archaic, it means old. So these live in extreme conditions, such as very hot, very cold, very salty. These lack peptidoglycan in their cell walls, just one way to tell between them. Here's the basic structure of a bacterium. So you have that circular DNA inside. There is an organelle called a ribosome that makes proteins so that the prokaryote can live its life. There are three bacterial shapes that we divide all bacteria into. Bacilli, which are rod-shaped, cocci, which are spherical-shaped, and spirilla, which are spiral-shaped. Bacteria have to reproduce because they are living, so they have to fit our 8 characteristics of life. So they go through what's called binary fission. Because they're very simple, they just make a copy of their DNA, split it in two, and the cells split in two. It's very quick, or it can be very quick, and they're very small, so it happens quickly. Another way that bacteria can exchange DNA is called conjugation. Two bacteria can link up with a pili that you saw in that structure, and they can actually allow little pieces of DNA to be passed between them. So this could be bad if they pass genes that make them more resistant to our drugs. Bacteria can also become spores. So when the environment is bad, so it's either too hot, too dry, they can go into a dormant spore stage, and that can last for months to years. So that would allow that bacterium to come out of the spore when conditions get better. Bacteria are very good for us. Our ecosystem would be completely different without bacteria because they decompose dead organic matter. So the trees and the animals that die, they need to be broken down. So yes, there will be scavengers that eat them, but there would still be too much laying around if we didn't have bacteria. So they recycle the nutrients into the ground. The other thing they do, which is really important, is nitrogen fixation. So nitrogen is a gas that we breathe in, but we can't really do anything with that nitrogen gas. It's bound in a way that we can't break it down. So what happens is the bacteria can take in materials that have forms of nitrogen in it, and the bacteria can change it, or what we call fix it, so that it can get back into the food web, and eventually we use it for protein. Here's another way that bacteria are good. In our food production, we use it. We make yogurt, blue cheese, wine, sourdough bread, among many other things with bacteria. In industry, they can use bacteria to digest petroleum in oil spills, so that's a newer thing that they do. They also use it in medicine. When we make drugs, a lot of times they use that idea of conjugation So they have little circular DNA plasmids, and we can insert DNA that codes for certain drugs into it, and the bacteria just make that drug and they spit it out in a form of gene therapy. Bacteria can be bad for us. The term pathogen is any disease-causing agent. So some bacteria are pathogens. So one way is that our cells actually become their food. So there's a bacterium called Mycobacterium tuberculosis, and that literally eats your lung tissue. That's the tuberculosis. It's a disease that you catch and you will always test positive for it. And it's around more than we think. People test positive, but their bodies can kind of hold it at bay and not show any symptoms. But then some people with a bad immune system, it reacts and they have a hard time. Other bacteria release toxins. So Streptococcus, if you've ever had strep throat, it can release toxins in your bloodstream. That picture is of a person's throat that has strep. So when the doctor looks into your throat, they're looking for something like that, or white spots in the back of your throat. Now, not everybody reacts exactly the same to strep, so it might not always look the same. So one thing that we use for these certain bacteria that are bad, you can use what we call a vaccine. We normally think of vaccines for viruses, but depending on what you have, they do have vaccines for some bacteria. So the vaccine is a weakened pathogen introduced into the body to stimulate the immune system to recognize a pathogen, possibly creating immunity. So one thing about vaccines that I think many people don't really know the proper way to describe them is that a vaccine doesn't mean you're not going to get something. What it means is that it's giving your body the ability to react to something quicker. So if you get a flu vaccine, your body sees the flu, those strains of the flu that are in the vaccine, and it makes memory cells for it. So then what happens is if the flu actually gets in your body, it can recognize them more quickly, and it kind of jumps ahead in the steps of your immune system to be able to react quicker. So it doesn't mean you won't get the flu, but it does mean that your body has a better chance of fighting it off so you won't get as sick. Some vaccines, like the chickenpox vaccine, usually means that you probably won't get the chickenpox. It depends on the virus. So when we talk about bacteria being bad, bacteria can be fought with antibiotics. So antibiotics are compounds that block the growth and reproduction of bacteria. And this is one activity where they have grown bacteria on this agar plate, and these little discs have a type of antibiotic that they've been soaked in. So you can see that there's areas that are clear around those discs, showing that the antibiotic is preventing the bacteria from growing. You can also control bacteria. One thing, well, we'll talk about that later. You can control bacteria by heat. So that's why you have a certain heat in your water when you use a dishwasher. You can, as long as you heat your dishes up really high, they will be disinfected. You can also use disinfectants. So that was a bad sterilization by heat. Disinfectants are chemical solutions that kill pathogenic bacteria, such as a soap or a disinfectant spray. Now, a lot of times we forget that you should spray the disinfectant on and let it sit for a minute to let that kill the bacteria, and then you wipe it down. A lot of times we spray and we wipe it away right away. And that's not the best procedure for killing the bacteria. We also use a refrigerator, low temperatures for our food. Low temperatures usually slow the bacterial growth down. So that's just one way to prevent its ability to grow quickly. We cook our food to certain temperatures depending on what bacteria are known to be in, like pork or certain meat. People like it at a certain temperature so that they know that it's cooked fully. Chicken, you don't want salmonella in your chicken. So you don't want to eat chicken that's pink unless it's been like smoked. That creates a different thing. So we talked about bacteria. Now we're going to switch to viruses. In 1935, American biochemist Wendell Stanley inferred that viruses are not alive.